All right, we're going to quickly chat about probability and inheritance. In the notes yesterday, you guys were learning how to use Punnett squares. And Punnett squares basically give us a nice way of showing how different sets of alleles can combine. So we could do each of these 5 examples. These are in your notes booklet. You could look at them if you wanted. A couple interesting ones would be something like number 2 here. So that was exciting. Try one more time there. So with number 2, we've got a big R and a little r, so a dominant and a recessive allele. And we're getting one of those from each parent. So here we're looking at the 4 possibilities. We could have a big R and a big R. We could have a big R here on the left and the little r up top. And we could have 2 little r's. And what this gives us here then is probability, right? The number of ways a given outcome can occur divided by the total number of possible outcomes. So for example, if we're looking at the probability of having a homozygous dominant genotype, that's this big R, that's 1 out of a total of 4. So the probability of getting this big R genotype is 1 in 4. On the other hand, if we're looking at the probability of showing the big R, in this case, if we're talking about the pea plants, the round phenotype, there we've got 1, this is a different color. Let's use a color we can see. 1 here, another here, and another here, because as long as we have one of the dominant alleles, we're going to get the dominant phenotype, and so there we'd have 3 out of 4. Interesting thing going on here, of course, though, is that both parents show the dominant phenotype, and yet it is easily possible, this little r, that they do have 25% of their offspring being showing the recessive allele. All right, so one of the things we can use, though, the idea of this recessive allele, is if we look at number 3 and at number 1. Number 1, all of the offspring here, I think it's pretty easy to see, will be round. There's no possible way for any of the offspring not to inherit the big R allele. So if I were to look at number 1, big R, crossed with little r, no matter which column we're in, we're going to get a big R. And by crossing anything with a little r and a little r, so this fully recessive individual, we end up with what we call a test cross. And if you're looking in your textbook, page, which one is this? I don't know. Page 604, questions 3 and 4 were some of the questions you had in your notes. And this is a fish breeder with these red male cichlids. Red is dominant to yellow. He must know whether the fish is heterozygous for these colors, suggest the way the fish breeder might find out the genotype of his red male. Use a Punnett square. And so what we're looking at there is we are looking at, well, if I have this red male, it's going to be big R and a question mark, and that's what I've got down at the bottom here. We're going to cross that with a fully recessive organism, so little r. In this case here, we're going to be looking at the offspring. Now, the neat thing with fish is you're going to have more than like one at a time. There's lots of offspring. And so depending on what this question mark allele is, what we will find out is that if this male is heterozygous, that is, it's got a little r allele where this question mark is, roughly, so if this question mark, I'm going to show it in blue, is little r, then we're going to have little r that we get from our fully recessive individual, and little r from the male. However, if this male fish has a big R here, what this is going to mean is our male fish is big R, little r, and all of the offspring will show the red trait. That's the big deal here with a test cross, is you're going to cross an individual with the unknown genotype with a fully recessive organism. And then we look at the offspring. Now, if this is some kind of a organism where they give birth to like one offspring at a time, this is a lot harder to do. You have to have multiple sets of offspring before this works. Statistically, you want to have a decent number. So with a fish, where hundreds of offspring at a time, that's going to work great. If we're talking about an elephant or something like that, that's going to have like one or maybe two babies every 2 years, this is going to be a silly and not super efficient way of determining what's going on. What that takes us is to a couple other patterns of inheritance. So one you need to know about is called pleiotropic genes. And this is where basically one gene affects a large number of phenotypic traits. So sickle cell anemia, which is a blood disorder, it's caused by one recessive gene. All right. So in normal individuals, they have what's called an HB for hemoglobin, HBA gene. Recessive individuals, they call it an H. This is one of the annoying things here. If they have two copies of the HBS and they don't use the proper set of lettering, or it should all be like the little letters for the recessive trait. But if somebody has two copies of this HBS gene, so the sickle cell hemoglobin, it's going to cause irregularly shaped hemoglobin molecules, and these are ones that lock together and they won't pass through capillaries. This causes weakness, fatigue, enlarged spleen, rheumatism, and it will cause all kinds of damage to the body as well. PKU is another example, phenylketonuria. It is caused by a mutation to the gene that codes to make an enzyme that in normal individuals converts phenylalanine to tyrosine. So that one gene is going to cause massive problems in various areas of the body. It will cause mental retardation, so huge changes across the brain. It'll cause reduced hair and skin pigmentation. And really the only solution to this is a diet that is free of phenylalanine. If we jump ahead, we can also talk about multiple alleles, and this is usually not a super difficult concept for people. Fruit flies have one of basically 4 eye colors, although I've seen sources that say there's actually many more than that. But we look at 4 types. Red is the wild type, and this is the most dominant. Apricot, honey, and white are mutant alleles. Drosophila melanogaster, the fruit fly, is diploid, which means only 2 of those 4 alleles may be present in any one organism. With multiple alleles, the genes are shown using an uppercase letter to denote the gene, and then a superscript number to indicate the hierarchy of dominance. And basically what this means is E1, this is the most dominant. This is dominant over E2, which is dominant over E3, which is dominant over E4. So wild type, E1 and anything else. Apricot, E2 and E2, or E2 and E3, or E2 and E4. Apricot, those ones there. Honey, E3 and E3, or E3 and E4. And white, the only way to get there is E4 and E4. So basically here we've got the first one. This is red. Apricot, honey, there's a little bit of color, and white would be just pure white. Incomplete dominance is another one you might see. In this case, we have two alleles that are equally dominant, and it's going to make a new phenotype that is intermediate between the phenotypes of the individuals homozygous for either trait. So basically, when we talk about incomplete dominance, you can see the way we show it. C, capital C, big letter R, capital C, big letter R, this is red, crossed with capital C, W, capital C, W, this is white, gives us C, R, C, W, and it's going to be pink. This is sort of like mixing colors in paint. If we look at something like codominance, this is where we get both alleles expressed in the offspring, but not in the same cell. All right, this is going to be HR, HR. We show codominance with small or with lowercase letters as the superscripted letter. And here we end up with all kinds of things we could look at. So roan cattle. Red crossed with white gives us roan. Roan is where any of the individual hairs will be either red or white. But there's going to be a mixture of those two that show up. When we talk about multiple alleles, we can get human blood types looking there. So A and B are codominant, and both are dominant to O. So you use a capital letter to show A or B, and then a lowercase letter to show the O. So again, more complex. The idea with really any of these questions, you need to know how to do a Punnett square. You need to know how to read the question, and then away you go. You can read through this bit here. Environment will influence phenotype. So water buttercups, broad, lobed, and flat leaves if the leaves develop above the water, thin and finely divided if they develop under the water. So same genetics, different environment will lead to different conditions. We'll look at dihybrid crosses later. For now, what I'm going to ask you to look at is your 18.3 pedigree charts. Oops, sorry, settle down there and jumped out of myself. So pedigree charts, these are going to detail a family tree, and they're going to show history of some genetic trait, usually a disease. And so you've got questions that I really want you to make sure are finished, and those are the ones in the 18.3 notes. Please work your way through those carefully. Discuss them would be a really good idea. And we will discuss them a little bit more on Wednesday, but my expectation is that you will know them and be able to answer those questions. All right, thank you.